Imagine a living thing so small that hundreds could fit across the head of a pin, yet so important that it helps shape the air you breathe, the food webs of the ocean, and even the dust that fertilizes distant rainforests. That organism is the diatom. Diatoms are single-celled algae found almost everywhere water and sunlight can meet: oceans, lakes, rivers, wetlands, damp soils, and even thin films of moisture on surfaces. They are not animals, and they are not bacteria. They are eukaryotic microalgae, meaning each cell contains a true nucleus and internal structures. But what makes them extraordinary is not just their biology. It is their scale of influence. Diatoms are microscopic, but together they form one of the great engines of the planet. They photosynthesize, grow quickly, build glass-like shells, sink into sediments, and help move nutrients through ecosystems in ways that are easy to overlook because the organisms themselves are almost invisible. The first key to understanding diatoms is photosynthesis. Like plants, diatoms capture energy from sunlight and use it to convert carbon dioxide and water into organic material. In that process, they release oxygen. Across the planet, diatoms are estimated to generate roughly 20 to 50% of the oxygen produced each year. That range is wide because ocean systems vary enormously, but even the lower estimate is astonishing. Every breath you take is connected not only to forests and grasslands, but also to floating microscopic algae in the sea. Diatoms are part of phytoplankton, the plant-like plankton that drifts through illuminated waters. They form the base of many aquatic food webs. Tiny grazers feed on them, larger animals eat those grazers, and the energy moves upward through fish, seabirds, and marine mammals. Their small size hides a planetary role, converting sunlight into food and oxygen on a massive scale. Diatoms are also famous for their shells, though scientists usually call them frustules. A frustule is a cell wall made of hydrated silicon dioxide, essentially a biological form of silica. In plain language, diatoms build tiny glass houses around themselves. These frustules are not rough lumps. They are intricate, often symmetrical structures filled with pores, ridges, patterns, and delicate geometry. Some look like discs or wheels, others resemble boats, needles, ribbons, fans, zigzags, or stars. Because their silica shells interact with light, they can produce structural coloration. This is why diatoms have been called jewels of the sea or living opals. The beauty is not decorative in the human sense. The structure helps the cell interact with its environment, allowing exchange with surrounding water while providing protection. A diatom is therefore not just a simple green speck. It is a living photosynthetic cell wrapped in a microscopic work of glass architecture. Most diatoms are solitary cells, but many form colonies. These colonies can take striking shapes: long chains, fans, ribbons, stars, or zigzag formations. Individual cells vary widely in size, from about 2 micrometers to as large as 2,000 micrometers. That means some are truly microscopic, while the largest approach the edge of visibility without magnification. Their forms are often grouped into 2 broad types. Centric diatoms tend to have radial symmetry, like a wheel or a circular plate. Pinnate diatoms are more bilaterally symmetrical, with a left and right side, often resembling elongated boats or blades. This difference is not just visual. Shape affects how they float, sink, reproduce, and interact with currents. In the open ocean, staying in the sunlit zone is critical because photosynthesis requires light. A cell's geometry, density, and colony shape can influence whether it remains suspended near the surface or sinks into darker waters. Diatoms can multiply very quickly when conditions are right. With enough nutrients and sunlight, a population can roughly double every 24 hours. This rapid growth happens mainly through asexual cell division. One diatom becomes 2, 2 become 4, and under favorable conditions an entire bloom can appear in a short period. But the life of an individual cell is brief, with a maximum lifespan of about 6 days. This fast cycle makes diatoms highly responsive to changing conditions. When nutrients such as nitrate, phosphate, iron, or silica become available, diatoms can surge. When nutrients run low, light changes, or grazing pressure increases, blooms can collapse. That rise and fall matters because bloom events feed ecosystems and influence carbon movement. Some of the carbon fixed by diatoms is eaten near the surface. Some sinks when cells die or are packaged into waste by grazers, carrying carbon toward deeper water and sediments. Silicon is central to the diatom story. Because they build silica frustules, diatoms draw enormous quantities of silicon from the waters where they live. Globally, they take in more than 6.7 billion tons of silicon each year. That is not a minor chemical detail. It links biology to geology. Diatoms pull dissolved silica from water, use it to build their shells, and after death those shells can sink. Over time, this contributes to marine sediments. In some places, the accumulation of diatom shells forms diatomaceous earth, also called diatomite. This is a soft sedimentary rock made largely of fossilized frustules. When crumbled, it becomes a fine powder with many industrial uses. It can filter water, act as a mild abrasive, absorb moisture in products like cat litter, and historically served as a stabilizing material for dynamite. A living microscopic algae cell can eventually become part of rock, industry, and human technology. The dead shells of diatoms do more than pile up under the sea. In some cases, they travel across continents. One remarkable example connects Africa and South America. Each year, the Amazon basin receives millions of tons of dust carried by winds across the Atlantic from the Sahara. A significant portion of that dust comes from ancient diatom shells, especially from the Bodil Depression in Chad. That region was once part of a system of freshwater lakes. When those lakes dried, they left behind deposits rich in diatom remains. Winds lift the dust and carry it westward, where it eventually falls over the Amazon. This dust helps fertilize rainforest soils. The connection is almost surreal. Microscopic algae once living in African lakes become airborne mineral dust that helps nourish one of the world's largest tropical forests. Diatoms are therefore part of a planetary transport system linking water, wind, soil, and forest. Diatoms usually move passively. Unlike many swimming microorganisms, most are carried by currents, turbulence, and mixing in the water. Wind-driven motion can keep them suspended and bring nutrients up from deeper layers. This passive drifting is part of why they are plankton, organisms that move mainly with the water rather than against it. But there are exceptions. Male gametes of centric diatoms can have flagella, allowing active movement during reproduction as they seek female gametes. Some diatoms also live attached to surfaces rather than freely floating. They can grow on benthic substrates, floating debris, plants, and other underwater surfaces, contributing to communities known as periphyton. So although many people imagine diatoms only as drifting ocean plankton, they occupy multiple lifestyles. They can float in the open sea, coat river stones, cling to aquatic plants, or live in damp soils. Their success comes partly from this flexibility. The inner biology of diatoms contains several surprises. Like plants, they photosynthesize with chloroplasts, but their chloroplasts have a different evolutionary history. They were acquired through endosymbiotic events, where one organism became incorporated into another over evolutionary time. Diatoms also possess a urea cycle, a metabolic pathway often associated with animals. In diatoms, however, it serves different biochemical purposes and helps them manage nitrogen metabolism. Some diatoms take symbiosis even further. Members of the family Rhopalodiaceae contain a cyanobacterial endosymbiont known as a spheroid body. This endosymbiont no longer photosynthesizes, but it can fix atmospheric nitrogen, giving the diatom access to a nutrient that is often limiting in aquatic systems. Other diatoms live in association with nitrogen-fixing cyanobacteria, including forms in genera such as Hemialis, Rhizoselenia, and Chaetoceros. These partnerships show how diatoms solve ecological problems through biological alliances. There is an even stranger evolutionary twist: Diatoms. These are dinoflagellates that contain diatom endosymbionts inside them. In studied examples such as Durinschia baltica and Glenidinium foliaceum, the endosymbiotic event appears evolutionarily recent. The diatom inside still retains many of its organelles and much of its genome, with minimal gene loss compared with older endosymbioses. In other words, this is a snapshot of evolution in progress, where one complex cell has taken another complex cell into itself and the relationship has not yet fully streamlined. The diatom endosymbionts in these dinoflagellates have lost their silica cell wall, making them the only known shell-less diatoms. That detail is striking because the glass-like frustule is usually the signature feature of diatoms. Diatoms remind us that even traits that seem defining can change when organisms enter new evolutionary relationships. Diatoms also act as environmental recorders. Because different species prefer different conditions, scientists can use living diatom communities to assess water quality. A water body dominated by certain diatoms may indicate particular nutrient levels, acidity, salinity, pollution status, or ecological change. Their silica shells preserve well, so fossil diatoms can also reveal past environments. Sediment cores from lakes and oceans may contain layers of diatom remains, each layer reflecting conditions from a different time period. By identifying species and measuring their abundance, researchers can reconstruct changes in climate, water chemistry, productivity, and ecosystem disturbance. This makes diatoms valuable in both present-day monitoring and paleoclimate research. They are not just passive organisms being studied under a microscope. They are data archives, recording environmental signals in their bodies and leaving those signals behind after death in sediments that can last for millions of years. Fossil evidence suggests diatoms originated during or before the early Jurassic period, roughly 150 to 200 million years ago. The oldest fossil evidence includes a specimen from the living genus Hemialis found in late Jurassic amber from Thailand. Since then, diatoms have diversified into thousands of documented species, with many more likely still undescribed. Around 12,000 species have been formally recorded, but the real number is probably much higher. Their long history matters because it means diatoms have survived major planetary changes while continuing to shape ocean chemistry and food webs. They are ancient, adaptable, beautiful, and functionally enormous despite their small size. A diatom is a single cell, but collectively diatoms help produce oxygen, feed marine life, cycle silicon, bury carbon, build sediments, fertilize distant lands, and track environmental change. The next time you think about what keeps Earth running, remember that some of the most important machinery is made of sunlight, water, and microscopic glass. If you found this helpful, please like this video and subscribe for more explainers.